So hello friends, welcome back to my channel Medicine of 101. Today we'll talk about one of the most important topic, that's the insulin receptors and the signaling transductions pathway. Okay, so how the insulin receptors are getting activated and what type of the signals will go from the surface of the cells to the intracellular level. That means it may go within the cytoplasm, it may go within the nucleus also. And what type of changes we'll see in these cases, let's talk about that. Okay. So when you are talking in terms of the insulin receptors, first we need to know that how the insulin is produced by the pancreatic beta cells. I have already explained in the separate video where I have explained in terms of the insulin synthesis and secretion. Okay, you can watch over there. Yet I'll just explain very briefly. See, insulin, what do we know? Insulin is the most powerful anabolic hormone. Insulin, the most powerful anabolic hormone. Along with that, we have to know also is the anti-catabolic hormone plus anti-catabolic hormone. I'll talk about these things, how it is working as the catabolism. Okay. Okay, before that, let's know how the insulin is getting secreted from the pancreatic beta cells. For example, when the insulin level that will get increased in the blood during the feeding states, whenever we have taken already the enough amount of the food at the time within the blood, or the glucose level that will be elevated. So whenever the glucose level that will be elevated at the time, or the productions of the insulin that will be also elevated. Okay, so how the insulin is getting secreted from the pancreatic beta cells, let's talk about that. So for example, we have taken enough amount of the food. So whenever the food particles, or we can say in terms of the glucose, that will be what? After getting here, that will be absorbed. So whenever it will be absorbed through the portal circulation, it will go to the liver, and from the liver, for example, it will go where? Within the general circulations. Okay. For example, it will go where? Within the liver through the portal circulations. So glucose, and from here we will get what? It will come over here, and it will be used by the hepatocyte cells, and it will be stored, and some amount of the glucose that will be released within the general circulation. So we will get the glucose within the general circulations. So then the glucose that will get within the general circulation. So within the general circulation we will get the glucose molecules, and from here it will go to the different tissues. One of the tissue, that is the pancreatic beta cells are also here. So if we talk in terms of the cellular level, the glucose molecules that will also go within the pancreatic beta cells. Now for example, I am just drawing the pancreatic beta cells over here. Okay. Okay, now see. Whatever the things we'll see over here, within the pancreatic beta cells there is the presence of the glucose transporters. Okay, so what type of the glucose transporter will get over here? There is the GLUT2 transporter is present over here. What is its functions? Its functions, whenever the glucose that will be reach over here, it will take the glucose and it will flip the glucose. That means it will take the glucose and it will flip the glucose within the cells. So whenever we will get the glucose that will be released within the cell, just flipping movement, then again it will go back to the normal form to take another molecule of the glucose. Okay. Now whenever we already got the glucose molecule, we know what type of the functions we will get over here. This glucose that will go under the glycolysis process through the multiple steps. It will go ultimately within the pyruvic acid. After that we will get the TCA cycle, where we get to the electron transport chain. As a result, we will get in these cases what? Productions of the ATP. We will get the productions of ATP. That means we will get ATP. Now, under normal circumstances, if we see within the pancreatic beta cells, there is a presence of one kind of the potassium leaky channel. Look, potassium leaky channel. So see, if I show over here, is the potassium leaky channel. Under normal circumstances, the potassium that will go outside. Potassium that will go outside, from the inside to outside of the cell. Why? We will see in these cases there are certain kinds of pockets. For example, this is a pocket where ADP can bind. Where ADP can bind. So whenever the ADP that will get bind over here, this channel that will be opened. But onset whenever we will get the productions of the ADP by the process of what? Glycolysis of the TC cycles. So whatever the changes we will see over here. See, in these cases we will get, for example, the ATP that will get bind over here, and it will block the what? It will block the, whenever the ATP that will bind over here. Okay, we can say the ATP sensitive what? ATP sensitive potassium channel over here. Okay, so the potassium that will be not able to go outside. As a result, the potassium that will be repelled back. So within the cell, the concentration of the potassium that will be much more. Whenever it will be more, the resting membrane potentials we will see the resting membrane potential that will be neutralized by the accumulations of the potassium ions. So as a result, whenever we will see the resting membrane potential that will raise to the threshold level, at that time, what do you know? The voltage sensitive what? Calcium channel that will be open. So whenever we are getting the voltage sensitive the calcium channel opening over here, there will be the influx of the rushings. There will be influx of the calcium ion. So whenever we are getting the calcium ion within the cells, at that time we will see within the cells there is the vesicles. Within the cells there is vesicles. Within the vesicles we will see the insulin molecules that will be stored over here. For example, let's show in this position. The insulin molecules that will be stored within the vesicles. Insulin means in terms of what? Insulin, C-peptide and little amount of the amylin also. Okay, little amount of the amylin. Now what the calcium that will do? The calcium that will come over here, and it will just take these molecules over here, and it will just do the fusions with the membrane. So calcium that will help to get fuse these vesicles. That means the insulin containing vesicles within the membrane. So whenever it will come over here, it will get fused with these regions. It will just secrete out the what? Insulin. So insulin that will come over here. So ultimately we got the productions of the insulin. And one more thing, whenever the ATP is binding over here, okay, whenever the ATP is binding over here, the potassium is going out. At that time, this voltage sensitive calcium channel that will be closed. So that will be workless. So in this cases, it will be closed in these conditions. Okay, it will not able to work. Onset of the productions of the ATP will get over here. The ATP that will be produced, then we will get this other things over here. Now, we'll talk in details about the whenever the insulin already go over, the insulin that is produced right now, and how the insulin that will work on the insulin receptors, and what type of the transductions pathway will get within the cells. Basically, that's the thing we will talk about right now. Now let's talk about who got the insulins over here. Before that, let's talk about this thing that what do we know? Basically, the insulin that acts on within the striated muscle. Within the striated muscles. Striated muscle means we'll get what? We'll get the skeletal muscle. We'll get skeletal muscles, along with that we'll get the cardiac muscle. Okay. Then also insulin works within the adipose tissue. And it also works within the liver. But the mechanism of the actions that will be different. In case of the, I'll talk in details. So in terms of skeletal muscles and the cardiac muscles and the adipose tissue, there will get what? The blood-mediated glucose uptake. But in case of liver, will not get these things, because liver is having itself what? The GLUT2 transporter, by which it can just take the glucose. But here the liver that will be stimulated by insulin by the other mechanisms, by some activations of some enzymes within the liver. That means the insulin that will help for the activations of some kind of the enzymes within the liver. As a result, we will see the liver that will be what? Excited to take the more amount of the glucose within them. I'll talk about these things later. Okay. Now let's talk about in details of the things. So let's talk about over here. Now, for example, I'm just taking the hypothetical cells. It may be the adipose tissues, or it may be the muscular cells, striated muscle cells. Okay, I'm just taking a single cells. Okay, from here I'll try to understand you guys. Now see, if I just say from here is the hypothetical cells. Whatever the things will get over, try to understand from here. Okay. So basically insulin what? It's a tetrameric protein. Insulin, that's the tetrameric proteins. Within the insulin will get what? The two insulin, the tetrameric protein. That means we'll get the two alpha subunit, two beta subunit over here. Okay. And the alpha subunits, they are extracellular. The alpha subunits, they are extracellular. And the beta subunits, there are some portions of the beta subunits, they are what? Some portions extracellular, some is what? Transmembranal, and some will be the intracellular. So if I show over here, and I think it will be easier for you to understand. Okay, let's show with this one, the alpha. So for example, this is the alpha subunits. Okay, let's show the more clearly. Is the alpha subunits. This to the alpha, and we'll get the beta also, but within this beta we'll get some portions. Try to understand from here. We'll get some portions within the beta. That will be the intracellular, some will be the transmembranal, and some will be extracellular. Okay, and if we just draw the membrane little bit clearly, so let's draw it more clearly for you. Okay, let's draw it, and I'm showing it again. Okay. Because the lipid, the membrane was the okay. Double membrane will get phospholipid bilayer. Anyway, we'll just think the same thing we'll get in this right side over here. Okay. Now first I said that we'll get the insulin receptors. So what the insulin that are already produced we got over here, how the insulin will work right now, we'll talk about that thing. Okay, so within the insulin receptors we'll get the two what? The alpha subunit and two beta subunits. So alpha subunits I have already said that will be the extracellular. So the alpha subunits will get over here. They will be extracellular. Okay, and what about the beta? The beta subunits, for example, this is the legs. So beta subunits, for example, they are what? Intracellular, transmembranal, and some extracellular. So whenever the insulin, this is the insulin receptor, so the two alpha and two beta subunits. So whenever the insulin that will come and that will get bind where within the insulin receptors, for example, they will get bind where first within the, they will get attached within the alpha subunits. So when the insulin that will come first where within the alpha subunits. So whenever we will get, try to understand, for example, these insulin molecules that will get attached within the alpha molecules. So first, if we write it over here in this sequence wise, so first alpha subunit that is getting attached with the insulin. So whenever it will get attached within the insulin, at that time we will get the transformation changes where within the alpha subunit. We will get the transformation changes within the alpha subunit. So we will get the transformation changes. So whenever we will get the transformation changes within the alpha subunits, try to understand from here, this same signal that will be sent from the alpha to the beta. So same signal that will be sent to what? From the alpha to the beta. And if we see the structures in between them, see, we'll get the, in between the two alpha subunits, we'll get the what? So disulfide bond, and in between them also we'll get the disulfide link over here. So we'll get the disulfide link in between the alpha and beta, and in between the two alpha we'll get also the disulfide link over here. Now, so whenever the transformation changes that will be sent, or same type of the signal that will be sent from the alpha subunit to the beta subunits, we'll get what? Within the beta subunits they will be also excited. So after that we'll get, in this case, the beta subunits, the beta chain over here you can say, that will be excited. Whenever it will be excited, whatever the thing we'll see over here, try to understand. Within the beta subunits, there is a presence of one kind of the enzymes. Now, the name of this enzyme, that is what? Tyrosine kinase. This is the what? Tyrosine kinase. Now if I show it over here, this is the tyrosine kinase. So what do you mean by tyrosine kinase? Definitely try to understand tyrosine kinase. Now kinase means the what? Adding of the phosphate molecules, and tyrosine means what? At the positions where the tyrosines amino acid they present over there. So wherever the tyrosine amino acid that will be present over here in these cases, in these same portions there will be adding of the phosphate molecules. And in these cases, whatever thing, the interesting thing we'll see over here, we'll get the cross phosphorylation. That means that whenever the tyrosine kinase, for example, this tyrosine kinase that will be activated, now this will just do the phosphorylation of the opposite one. And the same way, it will, whenever this tyrosine kinase that will be activated, it will just do the cross phosphorylations of the opposite one. So in these cases, we will do, here we get the phosphorylations. Okay, let's do in these positions, phosphorylations. Now, actually we are getting what? We are getting the cross phosphorylations. By cross phosphorylations, we are getting the autophosphorylations. By cross phosphorylations, we are getting the autophosphorylation. So what are the thing we'll get over here? This tyrosine kinase, let's write it over here. Tyrosine kinase that will be activated, and it will just do what? It will just do the cross phosphorylation first, then autophosphorylation. By doing the cross phosphorylation, it will do the autophosphorylation. So cross phosphorylation, by it ultimately we will get what? Ultimately we will get the autophosphorylation. So whenever these things will be autophosphorylated, these things will be activated. And whenever these portions that will be activated, that can just attract on top of the molecule also. And under normal circumstances, if the insulin is not bind over here, the insulin receptors that is inactive. Okay, at that time this will be also inactive. So one kind of the molecules will get within the cytoplasm. For example, so we will get this type of the molecules within the cytoplasm that is inactive. But whenever they are getting the phosphorylated over here, these portions they will be activated, and this will be also activated. Now these portions, or that means these molecules, or because of this protein, they can be attached within these phosphorus molecules over here, along with these phosphorus molecules. So whenever they are getting phosphorylated, we will see over here, these molecules, they will be excited and they can get attached within these portions. And what is the name of this one? What is the name of these molecules? That is what? Insulin receptor substrate. This is known as IRS. So IRS stands for insulin receptor substrate. So after autophosphorylation, we will get what? That IRS that will get attached. IRS attachment. IRS means what? Insulin receptor substrate. So whenever insulin receptor substrate will get over here, whatever the thing we'll see over here after that. See, within the insulin receptor substrate, there is also presence of the tyrosine amino acids, near about 20 to 22 amino acids. So what this tyrosine kinase enzyme that will also do the phosphorylations of the tyrosines over here. This tyrosine kinase that is present within the beta chains of the insulin receptors, that will also do the phosphorylations of on what? On the insulin receptor substrate. On the insulin receptor substrate where? Within the what? Tyrosine amino acids. So there are the presence of the what? Tyrosine amino acid, for example, the tyrosine over here, tyrosine molecules. Okay. Now what they will do over here, this tyrosine kinase, enzymes, they will just also do the phosphorylations. They will also do the phosphorylations of these portions also. They will do the phosphorylation, the tyrosine kinase also, of those tyrosines amino acid that is present over here also. After that, what type of thing we'll see over here? Try to understand. Whenever these things will be activated, that means we will get the additions of the phosphate along with the tyrosine molecules. At that time, we'll see from here, that means from the insulin receptor substrates to domain that will be opening. We will get the opening of the 2 domain. It will be activated and it will just open the 2 domain. In the same way, let me show in these directions. It will just open the 2 domain. For example, one domain, let's say the two domain. So whenever it will just open the two domain, what type of thing will see? Definitely this domain that will be the place for some kind of the what proteins over here. So what kind of the protein that will come over here? Before that, what is the name of this domain? What is the name of this domain? If I say there is a domain that is known as SH2, that is known as Sarc homology. Sarc homology. So Sarc homology domain. And this domain will be activated. At that time we will see there will be incoming of one time of the molecules. In the same way we will get the molecules. I will tell the name of the molecules later, but first try to understand the concept, okay, the sequence. Now whenever this molecule they will get attached within the SH2 domain, after that what is the function of this molecule over here, okay. So see, basically what do we know within the phosphate, if we just say over here within the phosphate membrane, phospholipid layers, what do you know? For example, is the phospholipid layers, okay, and we will get over here one type of the compounds we can get. See, one type of the compound we can get over here in this portions already. Okay, let's the name of the compound what is that? Is the phosphatidyl inosytyl diphosphate. Okay, inosytyl diphosphate. Within the membrane we can get this type of the structure. Let me show over here with the different color. Then I think it will be easier for you to understand. Okay. See. So if we see from these structures, what type of the structures we will get within the membrane, within the what? Phospholipid bilayer. Within the phospholipid bilayer we will get from here this structure also. What structures we can get from here we will get. Okay. These compounds we are getting over. This is known as what? Phosphatidyl inosytyl diphosphate, because already we will get over here the two phosphate that will be attached with these portions. We will get in the number 4 and number 5 positions of this inosytyl compound. We will get the attachment of the two phosphates already. Okay, it will already existed from the first time. But whatever the thing we will see over here, we will try to understand this. These enzymes or these proteins, what they will do, they will come over here and they will add. For example, we already got the phosphate over here. You will already got the phosphate over here in third and fourth positions. This protein, what they will do, they will add one more phosphate over within the third positions. Now, what is the name of this enzyme? It is adding the phosphate molecules at the third positions of the inosytyl compound. That's the name of this one. What? Phosphatidyl. Okay, if you write it over here, what will be the name of this one? Phosphatidyl inosytyl phosphatidyl inosytyl. Okay, 3 kinase. Phosphate. Okay, so if I write it over here in this way, that we'll get over here what? This is the name of this one, PI3. Okay. So PI3 that stands for phosphatidyl inosytyl 3 kinase, because it is adding the phosphate molecules at the number third positions or the number third carbon of the inosytyl compound over here. Okay. Okay. So let's continue. So we'll get over here what we'll get. See that phosphatidyl inosytyl 3 kinase. That means the name of this enzyme, the phosphatidyl inosytyl 3 kinase. That will just do the additions of the phosphate in the number third positions. So whenever we get the addition of the phosphate in number third positions also over here, at that time it will be activated. So whenever it will be activated, try to understand from here. Okay. So let's write it over here. We'll get. Okay, that means the PIP2 over here, that will be converted into what? PIP3, because try to understand, at first it was what? At first it was phosphatidyl inosytyl diphosphate. Whenever we are getting the activations of phosphatidyl inosytyl 3 kinase, that will add one more phosphate where within the phosphatidyl ionocycle diphosphate, or over here diphosphate. Ultimately we will get the conversions of the phosphatidyl ionocycle diphosphate into phosphatidyl ionocycle triphosphate over here. And what type of the activations we will get after that, let's talk about that thing. Okay, so whenever we get this type of the activations we will get, for example, well, I'm writing showing this one over here. So we'll get over here number 4th position, number 5th positions, and we'll get also the activations of the additions of the phosphate in number 3rd positions. After that, whatever things we'll see over here, try to understand from these questions. Whenever it will be activated, it will have some pockets. It will have some pockets to what it can just bind some molecules. There are some pockets that will be opened and that can activate some molecules. Whatever the molecules we'll see over here, try to understand from these portions. Okay. See, in these portions, when you are talking in terms of these things, that phosphatidyl ionocycle triphosphate, that means PIP22, PIP3 over here, whatever thing we'll get in these portions. Now, this molecule that is known as protein kinase B, this molecule that is known as protein kinase B, or it is also known as AKT. Okay. Now, what is the function of this one? This will just do these molecules, that means the molecule over here, that will help to get attached on more phosphate along with this one. Okay, that will help to get to get attached on more phosphate along with this protein kinase B or AKT molecules. Now, what is the name of this one? The name of this molecule over here. Okay, that is known as phosphatidyl ionocycle dependent kinase 1. Phosphatidyl ionocycle dependent kinase 1, the name of this molecule. Now, whenever we get, try to understand, whenever we get this molecule that will add the phosphate where within the PKB or the protein kinase B or AKT molecule, at that time it will be activated. And whenever it will be activated, it will give the multiple signals within the cells. I am not going in details, I'll just say, because in the next part I'll talk in details the rest of the parts. What are the changes we'll get over here, how they are acting over here. See, one of the signals that will come from the protein kinase B, or you can say the AKT, after getting phosphorylated, they will come over here, and we know where within the vesicles there are some certain types of the blood transporters. For example, where we are talking in terms of the adipose tissues or the muscles, when there is the presence of the GLAT4 transporters. So we'll get the GLAT4 transporters over here. So whenever we are getting the GLAT4 transporter over here, one of the signals that will come over here, and these signals that will help these molecules to get fused along with the membrane. So whenever we will get these things that is coming by the signals within the membrane, at that time we will get these blood or receptors or transporters, they will come along with this membrane and they will get fused, and what they will do, they can take the glucose. We will get the uptaking of glucose molecules. Okay. Other type of the signals that will come over here, try to understand from these portions, that will come over here, and these signals that will do many things in terms of the metabolisms. So if we say in terms of the metabolism, what are the things they will do? If we write it over in terms of the carbohydrate, in terms of the protein metabolism, in terms of the fat metabolism, what they will do over here. See, at first, the insulin, they will just convert the glucose into glycogen. We know what the glycogenesis process that will be stimulated over here. So, whatever thing we get over here, the glycogenesis. So, glycogenosis, this process will be activated. Now, for examples, we have eaten so much. Okay, for example, whatever the glucose that came within the circulations, from the circulation, whenever it will just go within the liver or the muscles, then for example, the glycogen storage full. There is no much more glucose that will be able to get converted into glycogen. Still, there will be some glucose that will be left. Okay, at that time, that type of the glucose molecule that will be converted into fat. Okay, so we can get over here also the fat. After converting all the glucose molecules to glycogen, still if we see there is remaining of some molecule of the glucose, then it will be converted into fat in the adipose tissues. And as you know, in terms of the proteins, if we say what the amino acid metabolism, especially we can write it over amino acid metabolism. Whatever the thing will get over here, this amino acid they will be converted into protein. And whatever the fat, this fat that will be stored in terms of the triglyceride over here, triglyceride. Okay, and another kind of the mechanisms will get over here, that is known as GRB. That means growth receptor binding. We'll get another kind of the domain over here, that is for the growth receptor bindings. From here we'll get the multiple signal that will come within the nucleus, and after coming within the nucleus we'll get the certain kinds of the genes that will be activated. Okay, and whatever the different kinds of the changes we'll get over here. Now another kind of the functions, for example, for this signal, whatever the thing will get over here, this thing, what it will activate the enzymes, the signals that is coming from the PKB, that will activate some enzymes. That means it will add phosphate on some enzymes that is responsible for increasing the what? Anabolic process. So the insulin hormones, what they will do by the intracellular mechanisms, they will activate the, or they will just do the phosphorylations, or they will just do the additions of the phosphate who are within some enzyme molecules. Then whenever these enzymes they will be activated, they are responsible for causing the what? Anabolic properties over here. And along with that, in the opposite directions, if we see these receptors or the signals will come in some molecules, they will also do the phosphorylations. And whenever they will just do the phosphorylation, this enzyme they will be activated. But these enzymes they will be responsible for decreasing the anabolic activity, or you can say they are responsible for decreasing the catabolic activity. So try to understand from here. It's very important concept that whenever we get the intracellular signals is coming over here, first they will just do the what? Mediated glucose uptake. Second, I have said whatever the thing will get the glucose that will be converted into glycogen, the amino acid that will be converted into protein, the fat that into what? In the triglyceride over here. And what about these things? That when another thing, there are multiple signals that will reach over here, and the signals that are responsible, basically they will activate the enzymes that is responsible for increasing the anabolic activity, and they will just depress, or they will just destimulate the enzyme that is responsible for catabolic activity. Okay, or they can activate some enzyme that is responsible for decreasing the catabolic activity over here. Okay, so this was all about in terms of the the insulin receptors and the signaling transduction pathway. The part one, the next part we'll talk about the signal that is coming from the growth receptor binding, and ultimately the signal that will reach to the nucleus. Whatever the clinical manifestations in terms of I'll talk in terms of some pathological conditions also over here. Okay, like the glycogen storage disease. Okay, diabetes, a little bit process of the diabetes also I'll try to cover over here. So this was all about in terms of insulin receptors and signaling transduction pathway. See you in the next video soon. Okay, if you don't get any point, please let me know in the comment sections. I'll read your comments and I'll try to help you guys.